Hello, everyone. Welcome to the Casual Causal Talk, Episode 8. Today, we have a very special guest, Adrian Olszewski. Adrian is a computer science postgraduate turned statistician, an expert statistician who has single-handedly cleared a lot of misconceptions in statistics. Adrian currently works at 2KMM as principal statistician and R programmer. And Adrian is also a part-time antique gadget repairer. Adrian, welcome to the show. How are you today?
Oh, it's welcome everyone. It's very great to be here. I'm very pleased to meet you finally, Venkat, after so many years of, meeting each other on LinkedIn. So finally we are in person. And when you introduced me, I just realized about my journey, that I said that I'm postgraduate turned statistician. So considering that I really disliked statistics in a college and I promised myself never again, and I just entered this field for just a few moments and stayed for 15 years. So it's very, it's
Yeah,
so strange to thinking about myself this way.
not a lot of people will believe this, but I think we are meeting each other for the first time, virtually also. In past 5 years, all our communication has been through LinkedIn and, a couple of emails were exchanged. You shared a couple of your excellent papers. And yeah,
It's
talking about your journey, right? You transitioned from computer science graduate to a statistician. That is a pretty remarkable journey. Can you explain a bit about your, career progress? Why did you transition from computer science to statistics?
uh, you know uh, the why was not made by myself. It was just by
Mhm.
accident. So
Okay.
after I graduated, I wanted to work as a software developer because this was my major, software developer. And I joined a IT company when I was a ordinary programmer. And one day, my boss, the CEO of the company, asked me if I had some elements of statistics, in a college, and can I recognize what is mean, what is median? I said, yeah, basically. Okay, Adrian, there is a little analysis to be done. Just don't worry about that. Yeah, and this is how it happened. So it turned out that this was analysis, my first in my life analyze statistical analysis in the field, in the area of clinical trial. It was an observational study,
Mhm.
and I quickly realized that after the college I have to uh, learn everything from scratch because we had lots of theory, but the theory was completely, unrelated to practical applications. A lot of theorems, proofs, lemmas, and so on, but there was very little practicing. So I had to start from practically from scratch, but this now differently. So when I was at the university, there was so many topics to learn, so it was a constant pattern to memorize, to pass the exam, and then to forget.
Mhm.
And it didn't it didn't result in great results. So here, to be able to apply the knowledge in my work, I had to learn properly. So
Mhm.
I had to mm, understand deeply what I do from scratch for every single thing,
Mhm.
for what does arithmetic mean. So basic things that I thought were just trivial. What can be difficult about the mean? Over time, it turned out that my understanding was very naive, and subsequent studies, subsequent projects made on real data, and in clinical biostatistics, the data are often well structured, definitely well structured, but very difficult. Clinical biochemistry data are very difficult, have lots of issues, quirks in them. So for every new topic, like a basic t-test to compare to means or the ANOVA and so on, I had to start from the basics. And now I had to do this in a different way to
Mhm.
understand and to use it the next day. So the transition was unexpected. It was completely accidental. And then after 2 years, I returned to IT. So I worked as a software developer for 7 years, then a software engineer. And in 2014, I returned back to a CRO company and returned to clinical biostatistics and stayed there permanently, I hope. So today, it started, just by accident, but today it's my way of earning money. And actually, I like it just for one reason. I always wanted to be a doctor, a physician. Medicine was of my great interest, but just by coincidence, I chose IT, computer science, but medicine was always someone back in my head, on my head. So here I can combine I learned how to like statistics, then how to love statistics, and how to combine it with medicine. So this is my story.
Yeah,
Actually.
really interesting story, Adrian. I think I can relate with your story primarily because, even though I did, my bachelor's in statistics, I wasn't that great. It was years afterwards when I had to, apply these techniques to real life problems, real business problems. I had to relearn, and I kind of, retaught myself statistics all over again. And that is where I got a lot of aha moment. Oh, maybe what I learned in the, graduation was not correct. Those things really help I think the contextual learning is what helps. If I can, relate both of a story, and if the audience is going to take away something from here, I think contextual learning always helps. And but anybody who is just graduating out of statistics, sadly, they may not have the context apart from the academic ones, right? You talked about CRO. Can you please expand what CRO is for the audience?
CRO means contract research organization, and this is kind of outsourcing. So when you have a pharmaceutical company who wants to introduce a new treatment, therapy, medicine, or independent researcher who is interested in doing some own research on the historically collected data, then they have two options. First, do it themselves, and pharmaceutical corporations usually have a huge departments of biostatistics consisting of dozens, hundreds of statisticians, but sometimes they prefer to outsource it to an external company and just to release, the resources for sometimes more serious or more demanding projects. So a CRO company does things, data management, statistics, sometimes supervising the process of clinical trials from the medical perspective as well. So CRO takes the tasks from the client, from the sponsor, because here clients are called sponsors, and we do this on behalf of the sponsor. So I do statistical analysis for many different clients, many different companies. This is what CRO companies do.
And if I may ask, what does your role entail? I remember sometime reading on LinkedIn, you had put a series of steps that your role entails. So if you could, explain about it.
In general, the scope of the places where a biostatistician manifests his, presence in clinical study is the places are just scattered across the whole period of the study. So ideally, in theory, we should assist from the very early stages of a study, from the study design. I would say this is the most important place for a biostatistician to take part, to join the study. When the study is designed, when the shape, the schema, the design, the necessary sample size, I mean the number of patients to recruit, when all this is just planned. Because if this process fails at this stage, the whole study may be just doomed, and you will realize this much, too far, too late to react. So many people think that statisticians are present just at the latest stages of the project of the study, just to perform this statistical analysis. actually we start, or we should start, because not every sponsor involves statisticians from the very early stages. So what I actually do, I assist sponsors in study designs, in writing the statistical analysis plan. This is some kind of black book of the study. There are two black books of the study: a protocol, which contains the medical description of the study, with just very elementary statistical chapters, a few sentences just, and the statistical analysis plan called SAP. And the SAP is the second black book from the statistical, very technical perspective, where every step of the statistical analysis has to be described, planned, and justified. Besides the basic plan, you need to have also alternative scenarios in case that something goes wrong. And usually all our plans are nothing but the wishful thinking. So we need to prepare not for the best, but for the worst. So if the assumptions fail, if the data are incomplete, or if something goes really wrong, we need to have backup plans. And all those things, hypotheses, statistical methods, tests, models, their properties, parameters, all this has to be planned. So the SAP. Then you need to design the deliverables. I mean the table shells, templates of tables, templates of figures, because actually this is the final product for the sponsor. Sponsors are not interested in math. They are interested in numbers and in figures. So my task is to propose the shape, the layout of the such tables. And sometimes it's, encloses in hundreds, dozens, tens, sometimes hundreds of tables. And after all, when we have this prepared, then I wait for until the data are collected. So when patients are recruited. But in the meantime, I write statistical programs. I'm preparing myself to do the final analysis, to not waste the time. And here we mainly use statistical packages that are based on programming to, achieve reproducibility in the analysis. So typically we use SAS. I personally use R. So I'm preparing the statistical programs or perform so-called interim analysis. So I mean, these are preliminary analysis before you reach the final one, the database log and the final analysis, just to see what's going on in the study. And after all, I'm writing the statistical analysis. I run the programs on the final clean data and write the statistical analysis report, which is the base for the clinical study report. And the clinical study report is submitted to the regulatory agency. So this is more or less, briefly speaking, what I do daily. So I plan, design, analyze, write report, program in R.
Yeah, that was a very nice breakdown. Talking about SAS in 2010, 2011, I started learning SAS because, I was maybe thinking to go into this pharma sector. And there was one company, who was ready to, take in the cost of SAS training. As you know, it's very expensive. I do have a SAS-based certificate, but somehow they ran short of money and nothing fructified. So I just stopped my knowledge with SAS-based, and after that, we didn't progress further. One of the reasons I started following you, Adrian, many years ago was because of the statistics knowledge that you espouse, and of course the statistical rigor that biostatisticians apply to their field, right? And somewhere I thought that if I could learn from you people, I can apply those same techniques into my field of marketing. But I always get a pushback. Marketers don't like so much of stats and math. Maybe I'm also accused of sometimes over sciencifying my field. But I believe that if you do the things rigorously, at least you will get accurate results. In your profile, I see that you mentioned the word frequentist. Why is it that the clinical trials domain is so heavily frequentist? Of course, I am brought in. I mean, I'm totally sold on that because initially in my career, I was kind of Bayesian. I call myself disgruntled Bayesian lover. Because I used to love Bayesian things, but when I started doing it, started applying it to real problems, I realized that the tool is simply not enough. So going back to the question, why do you think clinical trials domain is so heavily frequentist? And you may also, talk about the type 1 error guarantees.
Yep. So first of all, I put in my headline a frequentist just to tell people, tell the clients that, I usually don't do, I'm not experienced. I know there's the basics of the Bayesian analysis, but I'm not an expert in it. So just, just to save their time. Yeah, this is kind of information, not something that, not kind of religious approach to the constant war, I would say war, yeah, conflict between statisticians, Bayesians. No. The field in which I work, clinical trials, draws the best from both sides. So one cannot say that, oh, this is completely Bayesian or this is completely frequentist. In early phases, I mean phase 1 and phase 2A, with very, very few observations, very few patients, Bayesian analyses are normally used. The phase 3, which is for drug approval. So, this is the most famous people. Usually, when they say clinical trials, I guess they usually equate this with the phase 3 as the approval step. And here, actually, yes, the frequentist framework dominates, which doesn't mean that Bayesian studies are not performed. And there are even formal guidelines issued by the FDA, for example, for Bayesian. So this is not that we don't use such things. Also, study design, the step of study design sometimes also uses Bayesian approach to just limit the number of patients necessary to be recruited, just to cut the cost of the study. But at the core, clinical trials are a very specific process. Well, think about this way. You have several billions of people in the world, and most of the people take some drugs, some medicine, some therapy. I guess it would be hard to point to anyone who never, took even painkiller, right? So billions of people take medicines. They report quite often the result of this to their doctors, to physicians. So physicians, doctors have the feedback about how the medicine works. So this is kind of individual, little individual experiments. This knowledge is quite often stored in medical records. Pharmaceutical companies, they constantly, over and over, perform clinical trials, but they also get the information from physicians. They make their own research because otherwise they wouldn't do what is beneficial to do, right? So they do their research on what's going on in medicine, what kind of medicines are of best interest, in societies, what is best, and what works and what doesn't. Because if something doesn't work over time or is very toxic or have many adverse events, then this is a flag for them, a aha moment that maybe it would be beneficial to focus on something new, something better. Yeah, like we have new medicines, new drugs in cardiology, in psychiatry, right? For antidepressant, anxiety, and so on. So what does it mean? It means that little experiments are repeated over and over, which perfectly matches the idea of frequentism, right? So we have a set of repeatable studies. The second thing is that in randomized clinical trials, we work under randomization, which is, it's by definition, just by definition, sets a study that you work under the true null hypothesis. So by randomly assigning patients to treatment arms, and before the treatment was administered, you really work, which is very rare. Yeah, it's hard to imagine when and where actually we could work under true null hypothesis. In randomization studies, we actually do. This is the second thing. The third thing is that for some reason, ethical concerns, the regulatory agencies want us to, when we provide some statistical measure, some that quantifies the effect, the treatment effect. They would like to provide us some characteristics of the treatment effect in the long-term use, because this if we imagine that the studies are repeated itself many times, because every one of us becomes a little single small experiment, the regulatory agencies want to see if the actual work of the treatment isn't a sign of just, good luck, or that this working isn't a sign of bad luck. So we call this the type 1 error in statistics. And regulatory agencies want us to provide the long run false finding, right? Type 1 error, false finding characteristics. And in the frequentist framework, p-value, confidence interval, whatever we use, all those quantities guarantee that in the long term, the fraction of false findings, wrong misfindings, is controlled at some level. Typically 5%, it can be 1%, even less. It depends of the field, depends of the situation. This is a result of something that Bayesians somehow, throw against frequentists, that frequentism violates the principle of likelihood, because we take into consideration not only the actual data, but the whole range of unseen, data. This is conditioned on much broader virtual scope of data. But actually this very violation works for us here, because it also allows us to provide the long-term characteristics in terms of the type 1 error, or the family-wise error rate, FWER. What is interesting for Bayesian studies, there is a formal guideline which also requests the Bayesian studies to provide such characteristics, which may sound ridiculous, right? Just because this is Bayesian study, it's focused on the different things. So regulatory agencies say, okay, we have no issue, no problem with Bayesian study. Just remember to do some simulations. They propose a few approaches, do some simulations, and provide us with the expected long-run frequentist characteristics. So we can, this is important for ethical concerns. So, and the last, but not least, it's simpler. It's simpler in terms of some methods, because you don't need to, I don't know if this is how to say it to make it beneficial, because some people may say, you ignore the prior knowledge. No, it is not so. We use meta-analysis. We design the studies based on the previous knowledge. So this is not that the knowledge is ignored. Knowing what may be the minimum clinical important effect to be detected, we embed this information in our null hypothesis. So this is not ignoring any past knowledge, but definitely focusing on the experimental data. If you use some prior, and this prior will be very different from your actual empirical data, you will need to explain this to the regulatory agency, because it is okay that you use prior that is well justified scientifically, and it affects it makes the posterior distribution, drawing the means left or right, and it modifies according to your best knowledge. But if you choose a prior that is very different from what you got from your experiment, and either the experiment is flawed, or the knowledge you have was wrong, or you missed something, or something was spoiled, and this requires much more effort, and not always people are able to translate their knowledge into a good prior. So in frequentism, things go simpler at the cost of more complicated interpretation. But let me be honest. If I work under randomization, when there is a setup for causality, I would say causal scaffold, some framework for causality, by eliminating some confounding factors, then I don't really care what measures will be used to determine the treatment effect. It can be Bayesian, it can be Frequentist, it can be standardized Cohen-D effect size, it can be whatever, any number that has some property beneficial and accepted by the regulatory agency. So it can be, for example, some frequentist property, it can be some Bayesian property, anything. So this is only per agreement what measure we will use, because this measure, if it crosses some threshold of importance in this under this causal scaffold, this causal framework, let's say, then it's clear indication that this is something is really working, and this work isn't just a symptom, I don't know, of just pure randomness. We actually observe something. So the frequentist is historically the first here, has some properties that makes it beneficial. It agrees with the nature of repeatable, billions of repeatable micro experiments.
Understood.
And yeah, and has the properties that we actually are interested in.
Great. Yeah, that was an excellent explanation, Adrian. You talked about the misinterpretation of the frequentist methods, right? Be it confidence intervals, be it p-values. I think a lot of people transition to Bayesian methods because they find these methods little counterintuitive, at least the interpretation of it. But I would say that these methods were designed for scientific inquiry, or let's say clinical inquiry, which is not cut out for laymen, right? I mean, doing a lot of experiments and gaining evidence to refute your hypothesis is a little bit counterintuitive to people who are just trying to prove something from the get-go, right? So therein lies the difference. And I think some time ago, maybe it was last month, you penned your, I would call it Magnum Opus. I think the title was, "What is the role of frequentist framework, p-values, statistical NHST, and practical significance MCID in RCTs?" So much topics covered in there. If you would like to, go one by one, let's start with p-values, right? What is it?
Yeah.
Why do people get it wrong?
I really wouldn't like to scare our audience, because the p-value confidence intervals are, from one side, they are very often misused, and from the other side, there are some kind demonized, some like, don't touch this, you are not able to understand it. When I refer to the definition, then indeed it may be, the textbook definition may be twisted, but let's think about it. You assume that something doesn't work. So in terms of the randomization, okay, because this will set the real case of a true null hypothesis. So you have true null hypothesis. You know that the treatment was not yet administered, nothing happens. And this is your belief. And we also will use the, I believe this is Rubin's claim that to have causation, you need intervention, right? So you apply intervention, and now something happens. So you decide on what to observe. So you pick some quantity, some medical marker, for example, I don't know, the cholesterol level, hemoglobin concentration, or blood pressure, something like this, and you administer your treatment and observe what's going on. And you obtain you perform some tests, and you compare two arms, because in one arm is a group of patients, and in one arm you apply one treatment. It can be some currently used standard of care, some actively working treatment approved for use, or it can be placebo. So actually this is just fake treatment, just to make people think that they obtain something. Which one we use depends on the study, on ethical concerns, on what we are on the phase of study and type of study. The second arm is administered the experimental treatment. And now we apply the treatment, and for example, we agree to compare mean of the parameter we want to analyze. It is called endpoint. Clinical endpoint, the measure that we analyze, the medical biomarker. So we compare the means, and it turns out that the means the difference between the means is so big, so it created p-value that is very low. Now it means the p-value is a measure of, I can explain it in non-statistical way, as a measure of how much we are surprised by the data, assuming that nothing works.
Yeah.
So we have some assumption that nothing works. We collect the data. And if the data were compatible with this assumption, we would observe large p-value, which might translate to small difference between the treatment arms, between the means, for example. If we observe a big difference, so the p-value is small, this is surprising to us. This shouldn't happen if the treatment really didn't work. So this is a measure, kind of a measure of surprise, of being surprised, as I told you. Their definition says that under the true hypothesis, if we repeat the experiment many, in theory, infinite number of times, the p-value will be the probability of observing such difference as we got in every sample on even larger.
Yeah.
So if the probability of obtaining this is very small, so we say, aha, under null hypothesis, so under no effect, it's very unlikely to get so big effect, or even larger. This is just natural. If this big is unlikely, then even bigger will be even less likely. And when you repeat this experiment, you are guaranteed, under met assumptions, of course, that fake findings will not exceed 5% of all those samplings. And this is difficult to understand for an ordinary person because we don't think about an experiment like a repeatable process. But don't think about this way. Just focus on this. Low p-value, the data are much incompliant with the null hypothesis. So this is, we could replace p-value with any other measure, just agreeing what are the properties of this measure. If we accept those properties as convincing to us, we can use any other measure. P-value is just one of them in the frequentist framework. So I would say it's measure of incompatibility of the empirical data and the pattern that says nothing works. Confidence interval, I would say, not going into the statistical definition, is a range of all hypotheses compatible with our data. So if we analyze differences between two means and the confidence interval doesn't cover the 0 for difference or 1 for ratios, but let's focus on differences. If such confidence interval doesn't cover 0, it means that 0 is not a value compatible with our results. And this is exactly the same as rejecting null hypothesis. Because it says basically that our data are not compatible with 0 difference. And 0 difference was the null hypothesis that it doesn't work. So confidence interval is usually called by people, described as a range of plausible values. I don't like much the word plausible because it automatically converts to probability. And in the frequentist framework, we don't assign probabilities to estimates, rather to the process.
Exactly.
So the fraction, the percentage mark, is about the process. If we repeat this over and over and over, the confidence interval will cover the true value, the expected number of times, and only few of them will not cover this value. But I like thinking about p-value and confidence interval as a measure of compatibility.
Yeah.
What is the MCID? MCID is the minimal clinically important difference. Because now statistical significance has one more interpretation, at least to me. When we say that something is statistically significant, we have in mind that this is detectable. So you have some noise. some variance, and you have
Mhm.
a signal, some peak.
Mhm.
And if the peak is much enough exceeding the background noise, you are
Mhm.
able to detect it.
Yeah.
So it is kind of, detecting aliens from the outer space.
Mhm.
So if you have appropriately powered, sensitive detector, you are able to catch the signal, but you don't know if this is actually important. If this is a signal from aliens, or I don't know, your colleague just turned out on a microwave oven, for
Mhm.
example, 2 rooms or 2 floors below.
Mhm.
So statistical significance says it is statistically detectable from zero.
Mhm.
Statistically detectable from zero.
Mhm.
And if you have confidence interval that has no zero inside, but can be very close to zero, which means that compatible values can be of no interest for you as a physician, because you are not only interested in detecting differences, just any.
Mhm.
In medicine, you are interested in detecting differences that have some meaning. And I would call this clinical significance. So no statistical significance is just about being discernible, detectable, and clinical significance, clinical importance is of being meaningful, important from medical perspective. So if you have a drug that lowers LDL cholesterol, the bad cholesterol, right? You want the treatment that lowers this more than, let's say, 50 milligrams per deciliter. And this is your minimum amount of reduction
Mhm.
that is of interest, that is beneficial for the patients, for you, for the, I don't know, for the science and so on. And in clinical trials, we actually combine the two. So we want our treatment effect to be detectable over the noise. So we see that actually, yes, it is visible and should be meaningful. And we do this through by embedding the clinically important threshold into
Mhm.
the null hypothesis. And there are three types of trials, studies that use such, actually four, that use such knowledge. The first one is clinical superiority, which
Mhm.
means that one treatment must be better than the other, but by no less than the clinical importance.
Mhm.
So if you have a confidence interval, it will be easier to explain as a confidence interval, and you have
Mhm.
zero.
Mhm.
So in clinical superiority, you have zero. Then you have some, I don't have another pen, but okay,
That's okay. We
this is
can imagine.
zero.
Yeah. Yeah.
This is clinically important difference,
Yeah.
and this is your confidence interval. So the smallest value in the confidence interval, okay, let's think this is the smallest value, it must be greater by zero and even by the smallest clinically important difference. So
Mhm.
even the smallest compatible value in the confidence interval must be above the clinical importance. This is clinical superiority.
Mhm.
Standard superiority is to be above just zero. So it
Yeah.
only stays if one treatment, and sometimes it makes sense. For example, if you calculate not only treatment efficacy, but safety, and you compare the number of serious adverse events or number of deaths, for example,
Mhm.
then you cannot say that usually that above 5% we start to care. Sometimes we just want to know if that new treatment is worse because has greater number of serious adverse event by any amount. So then we use the normal superiority
Mhm.
above zero. Then you have non-inferiority. This is very interesting study because you allow you to compare two treatments and say, we would like the new treatment to be better, of course. But even if this is worse than the standard of care a little, but just a little, within some margin, we accept this. This will not work for placebo because the placebo does nothing. So you definitely don't want your new treatment to be worse than placebo, because it means that you are harming patients because they deteriorate over time instead of improving or doing nothing. But non-inferiority is typically used for studies for treatments that are already accepted in use, and you introduce a new treatment and you want to be better or no worse by some margin, within some margin of being inferior. This is acceptable. Why do we do this? Because, for example, your new treatment is easier to administrate. For example, you don't need to do any injections, you need to just swallow a pill. Or it has fewer adverse events, or is cheaper, has fewer bad reactions or interactions with different drugs. So there must be some other benefit. So you agree that you may be just slightly worse in what the drug actually does, but it must have another benefits. So then you agree on being non-inferior. And so we have three: non-inferiority, superiority, clinical superiority, and equivalence. Equivalence, it means that the new treatment is neither worse nor better. It's exactly within some range. So the two treatments are within some range of equivalence. And this is used for generic treatments. So when you have some, for example, patented formula, and then the patents, you found a new molecule, for example, and you just want to prove that your treatment, your generic treatment, is equivalent, is neither better nor worse. But to know this, you need to know the margin of equivalence, and this is the clinical importance. So the clinical importance in clinical superiority is the non-zero value that you must be better than this. In non-inferiority, the clinical importance is the acceptable margin of being worse, no more than this. And in equivalence study, this is the margin within you say that you are equivalent to the actively working approved treatment. So we have p-value, confidence interval, and MCID, which is the clinical importance. What was the other? Can you remind me?
Equivalence. I think it was equivalence. Equivalence. Equivalence test. You were talking about equivalence.
Yeah, equivalence. But I mean, what were the other terms that you asked me to explain? So we have p-value, confidence interval.
Yeah, I think we covered them all. P-value, confidence interval. I think MCID. Yeah.
Yeah.
I think
so
we have
in general, the role of all of them is to quantify the treatment effect. So first, if the p-value and confidence interval is just to say if this is statistically detectable, and the MCID is to say if this is actually clinically meaningful. And then we combine this into superiority, non-inferiority, and equivalence studies.
Yeah, that was excellent explanation, Adrian. I think that was a masterclass, I would say. But I would like to thank you because, we have taken a lot of inspiration, from the biostatistics field. We have also, applied superiority testing, non-inferiority and inferiority testing in marketing experiments. In fact, in one of our courses, we teach people how to do this. Of course, we have taken rich inspiration from biostatistics, how you guys do, the inferiority test, the superiority testing. It may not be to the same rigor as it is in biostatistics, but as much as it's possible, we are trying to do in marketing experiments. Thanks for that.
Very great to hear that this idea expands.
Yeah, great. So Adrian, we have been called many names on LinkedIn, statistical police and all that, but all we have been doing is clearing a lot of statistical misconceptions, right? I know a lot of your followers and fans would love to hear on this particular topic again and again. The topic is busting a lot of statistical myths, and the very first myth is logistic regression is not regression. Every time somebody writes it, I know for sure that you're going to show up. Like Batman shows up and he's going to save the day. I'm pretty sure that you will do the same. So I'm almost always correct that you do show up in such posts and clarify that myth. Yeah. So just for the audience, why do you think people have this big misconception that logistic regression is not regression?
There are just, I believe, only this week I found 5 or 6 such new topics, because on LinkedIn I search for a concrete phrase, logistic regression, plus despite, because this is part of, despite its name, it's not a regression algorithm. And then I sort the posts, from the latest one, from the newest, and then I observe what changed since my life, my last checking, my last search.
Okay, so that is the Batman call.
Yeah, exactly.
Okay.
I believe there are 2 reasons. First is that, and this is very sad to me, in machine learning, one of the most important tasks in machine learning is to predict, right? So to classify, to predict. So for numerical outcomes, we predict numerical outcomes. For categorical, we predict labels, groups, whatever we called it. So machine learning focuses on predictions, and that's perfect. So for numerical quantities, they use regressions, and they call it regression. Poisson, of course, the standard linear regression, Poisson regression, maybe, I don't know if any other, but I could name Cox regression, right? If we talk about survival analysis, gamma regression, beta regression, and so on. And for categorical outcomes, they use a family of logistic regression, standard logistic, binary logistic regression, multinomial, ordinal, and this trinity. From the statistical perspective, both linear and logistic regression are both about a very same idea, conditional expectation, because in frames of the generalized linear model, we focus on the conditional function of the data, conditional function of the data, conditional to the predictor, to the predictor. Yeah. And in the GLM framework, the function is expected value. In the general linear model, it's just arithmetic mean. It's just the conditional mean. So we estimate conditional means. In the generalized linear model, except for the trivial Gaussian case, which is the general linear model, the special case, we need to transform the expected values to stay linear with respect to the predictor. So we use the link function. So we say link transformed conditional expected value of self-distribution. And for the general linear model, the link transformation is trivial, identity, nothing is transformed. And the conditional distribution is Gaussian. For logistic regression, the link is logit function, logit, and the distribution is Bernoulli. And the conditional of the Bernoulli is just probability. So we stay within the framework of conditional function of the data, even being more specific, conditional expected value, with some meaning. It's easy. For logistic regression, it's just the probability. And in statistics, both of them are treated as regression, because here regression is just estimating the conditional expected value, or more generally, conditional function, which can be in quantile regression, you can have median or any other quartile. In Cox regression, you have conditional hazard. So I don't know, I cannot find any other. In a robust regression, you have some robust estimator of the arithmetic mean, of the central tendency. It can be a Huberized estimator, for example, right? So from statistical perspective, all those algorithms, all those special cases are in one just frame. In machine learning, people found that the fact that logistic regression gives you probabilities is an excellent ground for making predictions of categories, right? For classification. Yeah. So what they did, they used the regression, logistic regression, add one more layer, conditional rule. So you compare the probability versus some threshold, usually 50%, but it can be any other
Yeah,
depending on
0.5. Yeah.
I recall you wrote about this some time ago about choosing the threshold to match some actual problem, not just 50%, but in the simplest case it's 50%.
Yes.
And they obtain just two value, true, false, or one class, the other class. And instead of assigning a new name, logistic classifier, as they have for neural network classifier, naive Bayes classifier, they kept the name regression, but obtained classifier on the top of the regression. And now they have to justify before themselves and all others why logistic regression is not a regression, because it doesn't give you a numerical outcome, because it gives you just two classes, which is not true, because what you give two classes is the classifier built on the top of it. But another thing is, I noticed that some people asked me, Adrian, in linear regression, we have fractional numbers, some continuous numbers, and it produces such numbers. So this is understandable. And in logistic regression, how can you explain it? I say no. The fact that in linear regression, you also get fractional numbers, but these are means, expected values, not just the raw input.
Yeah.
And thinking this way, think about Poisson regression used for counts. On the input, you get counts. What do you get on the output? Fractions. Is this the same number? No, this is something else. Expected count, but it can be fractional. You can turn it into counts, you can round it, but you get fractional outcome, because expected value is by definition fractional, continuous number, continuous value. Same about, for example, Cox regression. You take two different quantities, survival time and the survival status. And what you get? Hazard, completely different quantity. So will you just stop calling them regressions? No. In terms of statistics, they all do the same thing. They estimate conditional function of some data. Logistic regression is no different. In machine learning, people just confuse the names. So instead of naming their algorithm logistic classifier, they kept regression. Now they have mismatch in the names. And the second issue is that people copy content a lot. So every time I see the post is one-to-one exactly of the same content, of the same shape, the same wording as before. And now, if you put lots of such posts, and then you train AI on it, for example, ChatGPT or Gemini or Perplexity, anything else, and all new people who join the machine learning read articles, and in each article they read that logistic regression is a misnomer, is not logistic regression, it's not a regression. Then AI is trained on exactly the same information, so it must give similar answer. Logistic regression is not a regression. People feel that this is something, that they found a mystery, something, I don't know, cryptic, and actually statisticians have no idea what they do, because in machine learning, logistic regression is not a regression, and everyone is repeating the same over and over. And I can see no way to break it, because now on even the best sellers in machine learning, I'm not going to call the book, you can easily Google it, which says that logistic is not regression. And this book is probably in most of the libraries of the top universities, at least maybe in Europe, maybe in Asia, maybe in United States, I have no idea, maybe everywhere. So if people see an authority that repeats the same, they don't even care that actual authorities like Cox, who actually, like Bergson, McFadden, Cox, Hastie, professors Hastie, Tipshirani, right? They wrote the elements of statistical learning. And both of them work on logistic regression and used it in biostatistics years before it was used for classification. And they have articles about how it is used for regression-related work. In the elements of statistics, they introduce it as a regression-based algorithm. They have never said it's not a regression.
Right.
And people call them authorities, which are actually they are actually authorities, but ignore what they say.
Yeah.
So this is this is repeating itself. So whatever I correct, and I can correct it even 1,000 more times, I'm afraid it will not go beyond LinkedIn, and this will. A few years later, we may be rejected during the interview if a data scientist will ask you, Venkat, what do you do at work? Well, I use logistic regression. For classification. No, for regression. What? You don't know what you say. Completely, sorry, rejected. You completely have no idea what you say.
Yeah, I mean, it is for this particular reason I don't reject any candidates, even if they utter, a wrong answer to this question, because I know there is so much misinformation out there.
Exactly.
Rather, what I do is, I point them to your post or mine and tell them, hey, you know what, please learn this thoroughly, but I'm not going to reject you for this reason, but at least you will learn this better. But coming back to your chat GPT and perplexity, I have a thought that, we must create a Galapagos Island where, all these original ideas are preserved and the chat GPT or any LLMs don't learn from it, right? At least that way we can preserve the accuracy and everything. But yeah, who knows? In the future, we may have to. And another thought that I had was the reason why people think logistic regression is not regression, it's something to do with the programming language. If you look at the Python syntax, there is absolutely no clue that you are using regression. But if you use R, if you look at the syntax, there's definitely a mention that, hey, family is equal to Bernoulli or binomial. At least you get to know that, oh, yeah, I am applying regression and it belongs to a family of Bernoulli or binomial. But in Python, that's not the case.
It's a brilliant catch. I noticed exactly the same. And it doesn't surprise me, because I know that many people in machine learning origin from computer science. And Python is especially popular in computer science, so naturally also in machine learning and AI. So if the only application of logistic regression in machine learning is to classify, then the libraries for machine learning implemented in Python go this way. So they don't care about all other applications. The R, for example, R performs quite the opposite. It goes from the statistical circle, and here I sometimes tell people I've never used logistic regression for classification. I had no reason for, because it's not a task in my daily job. While using it on daily basis, because I test hypotheses about treatment effect. When you compare the fraction of clinical successes between two arms. So you define somehow a clinical success, true, false, success, failure, and you compare the fractions between two arms. So you compare the fractions between arms or some contrasts of them. I mean, some more complex hypotheses adjusted for covariance. When you use logistic regression for assessing the missingness patterns. When you have missing data, right? We have three patterns: missing at random, completely at random, and not at random. So you can employ the logistic regression with the randomness flag as a response and check if there is some pattern through the logistic regression. So I use it for detecting some patterns in missingness. You can use it for implementing weighting algorithms like inverse probability weighting, for propensity score matching.
Yeah, we do it. Yeah.
So, yeah, it has plenty of applications, and classification is one of them, but not the only. So we cannot replace the overall quantifier with a specific one, right? So we just used one single application and equated it with as the only application of it. So whenever I say this, people in R using R say, okay, that's normal. we do the same. So the output of the function that calculates the general linear model is exactly what we need. We need the model coefficients, the standard errors, sometimes the covariance matrix, if we use repeated observations, and G estimation or mixed models. In machine learning, people need just classes. Maybe it returns some, parameters if you request them. So, but just by default, it focuses on a different tasks. So when there is a newcomer to Python and learns about the logistic regression, such person, they may just notice that, okay, just returns to classes, and never realize that it has any other applications, because in Python it does that. Of course, I know from my colleagues working with Python that it depends on the library that you use. But for example, scikit-learn, for example, it's focused on classification. So yeah, it comes naturally from the area of interest, I would say.
Yeah, I mean, people say that you must read the documentation, but hey, who really does that, right?
Who is documentation? Exactly.
Yeah, I think there was a big problem with the scikit-learn some years ago. I think it was in 2019, somebody found out that, under the hood, scikit-learn uses L2 penalizations by default for logistic regression. And nobody knew about it, and people are like, hey, at least you should make it explicit. And otherwise, people are going to, wrongly apply this technique. But people did not even understand what are they getting wrong, So it was again an educational process to explain to people, hey, why you should, be very vocal about what penalization that you're using by default and everything. But yeah, that's how things go, right? Adrian, I have a very interesting question for you. Is normal distribution the most prevalent distribution in the world?
What I believe people think that is most prevalent, but my opinion is that it's definitely important distribution for its certain statistical capabilities, properties. Yes, it naturally occurs in certain theorems like central limit theorems. When we perform inference, like Wald inference, this normality happens over and over, and you can find it in many places. Also, normality, I would say it this way. When you think about the world, the universe, and we can just temporarily, virtually think that the data that we observe are generated by some processes. Of course, this is, just a kind of mental trick, but I like thinking about this way. And if a process acts, if there are many processes and they contribute to something additively, then it results in symmetric distributions of data. So negative and positive values build symmetrically around the mean. And if you have a combination of many sub-processes, little processes that contribute to something, but contribute in same way, you can expect it will contribute negatively and positively in same way, you obtain symmetry. So and if the symmetry also has some value around, it concentrates, then the normal distribution may be actually a good approximation. Of course, we must highlight every theoretical distribution is virtual. This is just mathematical construct. No real data can be so. No real data, Gaussian distribution is not truncated. It spans from -infinity to +infinity.
Yes.
But if we agree on some approximation, like we do, for example, in calculus, right? We have curve, and we approximate the curve with a segment. So if the segment is sufficiently short, we can approximate the curve with a very small error, and we control the error. So if we agree on this error introduced by this approximation, it's all fine as long as it allows us to solve a real problem with this. So the normal Gaussian distribution is good for symmetric data with one peak, one mode, and not too big tires, because immediately people may talk about Cauchy, for example, and other heavy-tailed distributions.
Yeah.
But I just said additively. What about the multiplication, right?
Yeah.
In biochemistry, we have cascaded reactions. So one reaction cascades another reaction. We have a cascade of chain of reactions.
Yeah, a domino effect, yeah.
Domino effect, right. And this is, for example, enzymes or hormones. So one enzyme triggers another enzyme, enzyme, or one hormone combines with another hormones and so on. And this is why in pharmacokinetics, which is about what happens when you administer a drug, when you take a pill, what happens with the pill, you know, how it is administered, how it is distributed, how it is eliminated from your organism, right? And the concentration of the drug in your bloodstream usually is right skewed, just because, and related parameters are almost log-normally distributed, because cascaded reactions very often act multiplicatively. And such acting results in right skewed data, positive, zero truncated, positive, and right skewed data. So when we talk about normal distribution as the dominating one, we automatically exclude a big part of the universe, including living organisms, even bacteria, that where the multiplicative acting exists. Another thing, survival. And one thing, there was a scientist, Adolphe Quetelet. I don't know how to pronounce it properly. He was a polymath. He is known for inventing the BMI score, BM index.
Yeah, body mass index. Yeah.
Yeah, body mass index. Yes, he's known. He worked in statistics, mathematics. He was even a poet, as far as I recall, and he was a big fan of the Gaussian distribution. So he took the Gaussian error theory, and expanded this to the whole world. And he believed that every single quantity is distributed like the Gaussian curve normally. It was criticized by a person like Edgeworth, you know, the guy from Edgeworth series. Edgeworth.
Okay.
He said that that we need to distinguish a theory from real data, and this is not even a theory. This was just assumption of Quetelet, and we call this Quetelismus. Quetelismus is believing that the whole world follows the normal distribution. Nowadays, we know that, for example, we have mixtures of distribution. For some of them, we can separate the mixtures.
Yeah.
But some mixtures are inseparable because we have no knowledge about that, how to distinguish them, how to separate them. We just observe multimodal distributions. We may have truncated data. For example, age. Age is usually described as a Gaussian, as a normal one.
Yeah.
If it's in the middle. So this is 0 age, a newborn. This is elderly, and you are in the middle. So in the middle, it may be Gaussian. But if you go towards each end and consider things like the age of dying newborns or elderly because of some rare disease. So for newborns, it will be right skewed. For elderly, it will be, left skewed. But this is still age. Can we then say that age is normally distributed? No, it depends on the context.
Yeah.
Then you have survival analysis. In survival analysis, you usually observe much right skewed distributions. And it has even a scientific support, because we have something like Fisher, Tripet, Kolmogorov, or Fisher, Tripet, Gniedenko theorem, extreme value theorem, which says that if you take maximum values, if random variables, maximum values, and you standardize them, The distribution, if they converge in distribution, then they will converge to Weibull, Gumbel, or something else I don't recall. So it can be both. Weibull can be left and right skewed, actually. But in the survival analysis, if we analyze the maximum times, which are often very, very long, you naturally converge. And we know that Weibull and Gumbel are used in survival analysis, especially Weibull. Weibull distribution, right? In the parametric survival analysis. Accelerated failure time, for example. Another thing that just broke, my mind just exploded, is when I read a paper which called the log normal distribution a natural distribution. Why? It turned out that, for example, elimination of some substance from your organisms, it can be a medicine, for example, for majority of substances is eliminated at a constant rate, and this is described by a differential equation, and it's called first order kinetic equation. This is typically you can find it in radioactive decay, for example, a good example. And here it is constant, but in people, in humans, and in living organisms, can be even bacteria. You have the elimination at different constants, at different rates, depending on the treatment of your condition. I don't know, even your health condition and interaction with other treatments. So the rate of elimination differs. And if we treat this as a stochastic process, there is something called a Fokker-Planck equation, and it models the distribution of such changes. And it turns out that the solution to this equation is log normal distribution. So this is another reason why for pharmacokinetics we observe, this is not our guess, we just see this. All those concentrations, eliminations, all this follows log normal distribution. This is also why in pharmacokinetics analysis, we just routinely log transform our observations, because we know that what is the origin of this. But that's not all. The same applies to multi-agent systems. When we consider agent, like, I don't know, companies, people, agents like systems working, it can be a people, a person, a group of people, a group of companies, countries. If we consider them as systems and they exchange information between them and so on, it turned out, and this is described in this article, it also results in log normal distribution somewhere, finally. Then we have the Benford law. Benford law for example, numbers, right? Occurrence of numbers. Again, right skew distribution. So it turns out that also I read a paper about entropy in physics, that actual systems, open system processes that exchange energy and matter between them, also to the Fokker-Planck equation, somehow lead to the log normal distribution. This has physical ground. This is not only theoretical hocus pocus, a trick. This has physical ground. It was, really mind-opening. And what else? Where else we have so we talk about multiplicatively acting process, survival analysis, extreme value theorem, and one more thing. People believe that the central limit theorem leads to the normal distribution. They forget it's about the means, the distribution of the means. And some other estimators also can, through the certain theorems, can be inferred this way. But we need to remember that all those central limit theorems need some assumptions to work. It is not that you just, throw some at the input, you throw some distributions. They don't need even to be the same. They don't need to be IID. Not even independent, because we have some versions of the limit theorem that works also for dependent data. But for simplicity, let's focus on the standard Lyapunov, for example, Lindeberg-Levy, Lindeberg-Feller, Lyapunov, then they have some assumptions. The simplest assumption is for the Lindeberg-Levy, you just need to have finite mean and variance. But your variables need to be identically distributed independent. But for Lyapunov, you don't have to have identically distributed distributions. Only the higher moments, like third, fourth, higher moments, need to diminish quickly. So they are, these distributions are not distracted, right? They are not pulled in different directions. So the distribution of means actually converges to the normal one. The Lindeberg-Feller, or Lindeberg, just Lindeberg, also has an assumption that neither variance dominates about the others. So again, these assumptions are about providing quick and existing convergence in distributions to the normal distribution. What if this doesn't happen? We have problematic distributions like fat-tailed Cauchy, for example, Levy distribution, power law behaving distribution, for example, Pareto distribution. And this, okay, one may say that we have more generalized theorems like Kolmogorov-Gniedenko. And this theorem tells you that if you have distributions that are, how would it say, if there is a property that they addition, you add them, you get the same stable. They are stable under addition. For some stable distributions, like they can be fat-tailed. They may have problems with variance, but they are stable. So they sum leads to the same kind of distribution. Gamma is stable distribution. Gaussian is stable distribution. Levy is stable distribution. Unfortunately, Pareto is not, but Cauchy is stable distribution as well. And the Kolmogorov-Gniedenko says that if you apply this to difficult distributions where the normal limit theorems may not work at all, I mean the convergence may be very low or not happen at all, it will lead to a stable distribution as well. Not necessarily normal. Actually, it won't be normal, but will be stable. But that's all. So even if we take the most general theorem, we are not guaranteed to see normality. And yet people forget about the assumptions and they say, central limit theorem, regardless of which, either will make it normal. And then they say, you just need 30 observations to get it normal.
N greater than 30,
Yeah, and I like Professor Rand Wilcox about in his books about non-parametric methods. He said a very wise thing. He focused on the T statistic, from the T test, on the T statistics. And he said, you forget one thing. You have the nominator and the denominator. What you want for proper inference is that the whole statistics must follow some distribution, the T distribution, the student distribution. You have the nominator, and just for means, it may be quite easy to have the convergence even for 5 observations. I saw such simulation 5, and it works. But you also have the denominator, and the variance is especially sensitive to problems with normality. And so it may happen that even for 300, for 500, the convergence will be slow. And Professor Frank Harrell, I believe he is also much recognizable person in statistics. Professor Harrell provides an example when he samples from log normal distribution. 50,000 observations was not enough to make appropriate coverage probability for a confidence interval that he calculated. Of course, one may say, Adrian, you missed the point. You misapply a statistical tool to the problem. And I fully agree. People believe that central limit theorem will, make the dirty work, will do everything for us. but they don't ask the basic question: is arithmetic mean actually a meaningful measure for what we need? Because then, yes, you can take 1 million of observation and the limit theorems, one probably will work then, but you will obtain technically proper answer to a meaningless question. So this is relying, and people do simulations. And what people use for the simulations often? The exponential family of distribution. And you know what? It turns out that for the just use the word exponential family of distributions, it worked perfectly for very few observations. But let's try with the fat-tailed distribution or much skewed distribution. And it doesn't go easily this way. So first is about the assumptions. The second, it pertains to means. And the third topic is that for many processes that are not built by additive processes, not resulting in symmetric distributions, relying on the CLT is just pointless. This is misapplying the tool to the problem.
Exactly.
So to conclude, Gaussian distribution is important for many reasons, but definitely I don't think it dominates in the world. And for example, the log normal distribution is one of even it can be inferred from natural physical processes, and many other distributions, empirical, not just theoretical, but empirical, don't follow symmetry. Symmetry is not the only pattern in the universe.
Yeah. Again, a great explanation, Adrian. And if I may just add, there was another myth also associated with CLT, that it starts to kick in at n greater than 30. But I think that myth arose because, in my statistics days, we used to use something known as the statistical tables. And for each distribution, we used to have the degrees of freedom and the statistical significance. We used to look it up.
Yes.
And that ended at n is equal to 30 for T distributions. And people had to fit so many distributions into that small book, because of that, they had to squeeze it to n is equal to 30. And people thought that, oh no, after n is equal to 30, T distribution becomes magically like a normal distribution. And that was the, source of the myth.
Because in statistics, 30 is infinity.
Yeah, you could say that. Yeah. Yeah, coming back to normal distribution, because of this particular myth, people go wrong in so many ways. I have interviewed people, and in my line of work, which is the market mix modeling, when we have independent variables, they think that independent variables need to be normally distributed. But because MM is just a glorified, I would say, multilinear regression, it is not about the individual variables. It is the error which has to be normally distributed, so that you can draw meaningful inferences, as you talked about. But yeah, that lends to another, series of myth. People go wrong just because of this particular myth that, hey, I can see normal distribution everywhere. There was this famous church, I don't know whether it's in Poland or Iceland, which is the shape of a normal distribution. Is it in Poland?
No. But I remember the church, yeah.
Okay. Okay, yeah. Yeah,
the
so
church
somebody,
of the normal distribution.
yeah, it has become like that. I don't know, it has a proper
Yes.
name, but I think people are like, hey, no, it is the church of the normal distribution, and every statistician wants to make a pilgrimage there.
Yeah, absolutely.
That's very funny. Yeah, talking about, transformations of, because people think that something has to be in a particular distribution, they log transform it, right? How, problematic it could be when you do log transformation. Maybe we can just take a simplistic example of linear regression.
Yeah, transformations, and I'm glad we start from log transformation, because there is a topic of box Cox and even more general transformation that goes even more difficult. This is a tool that, like any other tool in statistics, has its use, and I'm definitely not against that if done properly. Now, if we focus on the regression, and this is difficult to imagine, so let's start from let me explain in a minute just why I'm doing this, just to introduce the problem. Let's imagine a model for ANOVA. So we have just one categorical predictor. So when you fit a linear model on this, so that is actually this is linear model used for ANOVA. This is not ANOVA. This is used for ANOVA. Another thing. Then you estimate group mean, right? Group mean, and in every level of the categorical predictor form groups, distinctive groups. In every of this group, you have some distribution. Ideally, this should be normal for general linear model. And this translates also, because I know that some people may say, Adrian, no, this is about residuals. For ANOVA, this is exactly the same as checking the per group distribution, because, if you subtract the mean from each group, you will get centered data, centered around the group mean. And if you stack them together and they have the same variance, then you get the residuals, because residuals are the actual data, predicted data minus the data, actual data minus the mean, predicted value, minus the prediction. And the mean is the prediction here. So actually, we have some per group distribution, we have some mean. And now let's see what a person may, let's imagine this. I will have 3 groups. The first one, and all of them will have the same variance for simplicity. The first one has mean 1, the second is 2, and the third one is 5. So we have 5 Gaussian distributions. When we observe the response variable, the y variable, the response, right?
Yeah.
If you put it in a histogram, you will see a skewness. Skewness as hell. It will be right skewed. But I could make it differently. I could make the first one mean 1, then 7, 9. So you have left skewed distribution. And people say, I must transform this somehow to make it normal. First of all, what is necessary is definitely not the normality of the response. Neither the response nor the predictor needs to be normal. The residuals needs to be normal. And you can imagine it this way. Take a log normally distributed predictor, continuous predictor, add some Gaussian noise, and obtain the response. Also log normally distributed. And you see log normal on the input, log normal on the output, and the Gaussian noise added between them. So the residuals are normally distributed, despite having predictor and the outcome log normally distributed. So this immediately illustrates why we don't need normality in neither predictor nor the response. But going back to our example, people look at the response together and they forget that this is a mixture, not sum, a mixture of the per group distributions. The group coming from the categorical predictor, yeah? So the levels of the predictor form groups. In each group, you have some independent data. And when you look at it mixed together, you observe a skewness. But individually per each group, they are perfectly normal. And this is actually what matters. If you think about normal regression, I mean normal with a continuous predictor, it's more difficult to imagine that that this is about conditional distributions per each value of the continuous predictor. If you use Google and type regression conditional distribution and switch to images, Google Images, you will see the perfect illustration of that. So you will see those conditional Gaussian distributions per each value of the X predictor. So actually, the response is the conditional mean per each value here. And now, if you look look at the mixture of those, this also may look skewed. And when log transformation may work, when may it completely not? If the smaller the quantity, I mean, the smaller the intervals on in the predictor, because for categorical you have discrete, right? Discrete predictors. Yeah. They form individual groups. And here you should look into each group and assess the distribution. If they are normal, you don't touch it, because what you observe for the response is just a virtual distribution, is a mixture, and you don't care about that. For the linear regression, where you have continuous numerical values on the X predictor, it is more difficult to assess. Actually, you have no way to assess. You actually see that that the response is skewed. So you need to assess the residuals. If log transforming helps the residuals, it means it was fine. If it doesn't help to the residuals, it means it wasn't necessary. The same with the categorical predictor. It may happen that if you had 3 perfectly Gaussian per group distributions and you log transform everything, then you artificially skewed, you destroyed, you damaged the per group distributions. So the mixture, the observed mixture, the response variable may be symmetricized, it may be more Gaussian, but at the same time, you spoiled the conditional distributions, and the residuals may be horrible. So the conclusion is, first, when you log transform the response, ask yourself a question, because what does it mean to transform the response? On the other side of the equation mark, you transform also everything, including the errors. Now they are multiplicative. You assume that by transforming, you go from multiplicative errors into additive errors. So you need to answer a question. By log transforming the whole response, do I expect that the model describes multiplicative process? Is it meaningful? And the second question, why do I do this? Some people do this to stabilize variance. Despite we have, but there is the generalized linear model. It does something different. It transforms not the raw observed data. It transforms the expected values to put them on a line, right? Linear to the predictor, to make it linear. Expected values, conditional expected values. And it doesn't touch the error, the distribution of error. So if you one needs to make this distinction between transforming the entire model, log transforming the entire response, entire model, believing it describes some multiplicative process, or use the generalized linear model. When you don't focus on the error structure, you actually, and you are guaranteed that you don't just transform data, you transform the expected values. This is what is of interest. And most importantly, observe the residuals to learn if the transformation makes sense. And the last thing, because the transformation changed something, right? If we, for example, if you have t-test, just compared to groups, t-test is a kind of it has a linear model under the hood, general linear model, just with a single binary predictor, two-level predictor, general linear model. What happened when we log transform the data? We actually now analyze not the difference between arithmetic mean, but the ratio of two geometric mean. Here it was interpretable. Now, if you try to back transform the confidence interval, for example, you may get some bias because of the Jensen's inequality. And Jensen's inequality says that transformation of expected value of something is not the same as the expected value of transformation of something, unless the transformation is trivial, identity. So this way, one needs to carefully think, what will change when I transform? I will change the mean variance relationship. I will change the hypothesis that I will test. And let's pray that the transformation was meaningful. So I mean, when you have array squared meters, the square root will turn it into lengths. Okay, maybe meaningful. If you have volume in cubic meters and you take the root of the third grade, cube root, yes, you will take length again. You can apply the reciprocal transformation, maybe to velocities and so on. You can apply log transformation to multiplicative data to switch to additive scale. These are meaningful transformations, but what will happen if you take box Cox and it will give you the coefficient like minus 0.67?
Yeah.
This is not close to any meaningful kind of transformation. And what hypothesis now you are now assessing? How did you change the mean variance relationship? How did you transform the errors, the distribution of the errors in the model, and make sure that this is still interpretable? Because it, for predictions, it may be perfectly okay. You just want better prediction. So you can make a black box, add stack transformations as long as your predictions are good. You don't care about the technical. In my field, I care about the interpretation, about bias, about interpretation. Yeah. I must justify the transformation. And the formal guidelines in my field say, if you need, you can transform, but don't apply blindly transforms just because, you observe something and you make the decision to do this, and never transform the data just by looking only at the response, because it may happen that the conditional distributions were perfect and what you observe is just a mixture of them. And by transforming, you will spoil these conditionals. So this is not trivial at all.
Yeah, another great explanation, Adrian. Coming to another myth, right? I've seen this quite often, that non-parametric tests are better than the parametric ones. I mean, firstly, people don't understand that when they're telling non-parametric, it doesn't mean no parameters, but many, right? That is the first step people don't understand. Have you come across this particular myth or understanding that people think that non-parametric tests are better than parametric ones just because they have done away with certain distributional assumptions?
Yes, I quite often see the claim that non-parametric means no problems, because problems come from the parametric assumptions, and since the parametric tests or methods use theoretical distribution, and actually we never observe parametric theoretical distribution, so get rid of those and use, for example, rank-based methods like Wilcoxon, Cross-Wallis, and so on. So, yeah, this is not easy to explain, but let me start somewhere. First of all, why do we use parametric methods? Because by making some reasonable assumptions, it all starts of how reasonable are your assumptions. So again, if I know from the domain knowledge, from past studies, from my experience, from my educated guess, whatever, that some quantity can be, with some error, of course, approximated well in population by the Gaussian distribution, very good, because this distribution has just two parameters. So if I also fix the variance, I can only focus on the shift in location. So I can just compare means. This is easy, and this easy is reasonable easy and meaningful, as long as I can justify such approximation. Of course, I can use any other theoretical distribution with its parameters, as long as I can interpret them. So parametric maybe makes some assumption about a theoretical construct, but as long as it's meaningful, as you can justify it, and the error committed this way is acceptable to you, you get meaningful interpretation. Now, just assume that you assumed some parametric distribution, some test, for example, using Gaussian distribution, but you performed some testing and the test rejected normality. First of all, I don't want to, multiply topics. I will just only make a little digression that I really don't like testing normality through formal tests. Just to our audience, there exist about 90, I know because I counted them, 90 tests of normality or non-normality, because we cannot prove the null hypothesis, right? We rather check the divergence from the normality. and each is focused on something different. So some tests look only at the higher moments, like skewness kurtosis, like Jarque Bera test. Some tests look at some correlations with some scores, like Shapiro Wilk.
Wilk Shapiro.
Cumulative distribution functions like Kolmogorov-Smirnov and its improvements, Kramer von Mises and Anderson-Darling. So they focus on different aspects, and it may easily happen that one test will detect something that the other test will miss completely, even Shapiro Wilk. I like an example where I prepared such a distribution which has two modes. It looks like a Gaussian with a hole in the center, perfect hole. And I did this in a way so the skewness is zero, almost zero. This is perfectly symmetric, and the kurtosis is equal to 3, or the excess is equal to 0. So the tests that look at the higher moments return p-value almost 1, like 0.9999 and so on. While the Shapiro-Wilk also had problems, but for example, Anderson-Darling rejected at very low p because it was able to detect this very kind of issue. So first you need to say what kind of non-normality test you used. Another thing is that at small samples, when we need those tests the most, they horribly lack power. Because having just a few observations, those observations match practically any distribution. You cannot clearly say which one it matches the best or from which it differs too much. And when the sample size increases, those tests gain power horribly. And this is good because actually they want to assess how your data diverges from the theoretical pattern that you can never approach. But what it means that even for visually indistinguishable differences, perfect for the central limit theorem to work, like in the Wilk inference, likelihood ratio test, Rao score, perfect for this. But the normality test rejects the hypothesis. And what we are going to do? The textbooks say, if the normality test rejects the null hypothesis, switch to non-parametric methods. And now, while the conditions may be actually perfect for the parametric one, but we automatically do something different. But what is the risk? Non-parametric methods have, again, like normality test, shape a few families. We have rank-based methods like Kruskal-Wallis, Wilcoxon, right, Friedman. We have quantile-based, like quantile regression, including the test of medians, for example. I'm sorry, just a digression. Mann-Whitney, Wilcoxon, or Kruskal-Wallis, they don't compare medians in general. There's another misconception. And we have also resampling tests, like permutation, bootstrap. We can have a kernel density estimation, and so on. And people typically, speaking about non-parametric tests, think about those rank-based. And the rank-based tests are can have very difficult hypotheses because the general hypothesis is, the null hypothesis is stochastic equality or equivalence. The alternative hypothesis is stochastic superiority. What does it mean? Stochastic superiority means, in brief words, that if you have two groups, A and B, and now let's assume that you imagine that you connected pairwise all observations from two groups. And then you calculate how many pairs were increasing, how many were decreasing, and just calculate the percentage of the increasing ones. If it's about 50%, above 50%, above, then it means that the observations in the group B dominate, so are statistically larger than the observations in the group A. If you have 50-50, so if this 50%, it means that neither group dominates over the other. Seems pretty easy, but this doesn't map to means nor medians in general. It will map to medians if both compare groups are of the same shape. So it can be skewed, but must be skewed exactly in the same direction. It can have multiple modes, but both must be exactly the same. And there is even stronger assumption that also those need to be symmetric, not only of the same shape, but also symmetric to approach medians.
Mhm.
And this is quite strong assumption. Most of the time we use non-parametric tests because the distributions are, completely chaotic, of very different kinds. And this is so what may happen? It may happen that you will have perfectly equal medians. Just exactly, not approximately, exactly equal medians. And the Wilcoxon or Mann-Whitney, just another version of the test statistic test, will reject p-value at very, very, very low significance level. So it will be just 0, 0, 0, 0 for quite not a big sample. I don't know, 30, 50, not a just big sample. It won't be a matter of sample size. So to what it was sensitive? It was sensitive to what actually, if not medians? Well, maybe to means, maybe to dispersions, maybe to shapes, maybe to everything. So as long as you cannot make two distributions same distributed, at least regarding the dispersion, so the variance and the shape, because if they are IID, independent and identically distributed, they can differ just by location. So this is good. But as long as they differ in some other way, you cannot attribute the difference reported by the test to any concrete property of this distribution. So it may happen that the test will reject for equal medians and even for equal means, not together, not at the same time, but either this or this, and the vice versa. You may have very different means or very different medians, and the p-value will be almost 1. And I prepared once illustrations you can find it on LinkedIn, where I showed both cases. So in one case, the medians were identical, but the stochastic superiority was about 70%. So the test detected it, despite equal medians. And in the other case, I made the data, made the data in some specific way. Half of the data dominated, in both groups, the data dominated in opposite directions. Half the data, in one part, it dominated over this other group. In the other half, the other group dominated. So they cancel out. But the medians were very different, and means as well. What does it mean practically to us? If we wanted to compare means, originally, we want to focus on means, because this is meaningful, because everyone else does it in the scientific literature, and we want to be compatible, and we wanted to compare means. Then it turned out that the assumptions weren't met, they failed, so we switched to rank-based method. Now it turns out that we obtain something very, 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 very unexpected, because this test, the rank-based test, tested completely different hypotheses. So one may say, okay, Adrian, if do we actually need rank-based method? Yes, we do. For example, if we have ordinal data and the distances between the ordinal levels are not equidistant, so there is no constant unit between them. Like on the Likert scale, right? We can have items numbered 1, 2, 3, 4, 5, but the distance, meaningful distance between 1 and 2 is not the same as 4 and 5, so there is no unit. So here there is no algebraic operations, there is arithmetic operations defined, so also the mean is meaningless. But if they are equidistant, like on the visual analog scale, when a patient points on a scale from 0 to 100 about the pain, this is something that can be well approximated with an ordinal scale, and then no problem. But when we have ordinal data in general, we can only say that some observation is larger or smaller than the other. So here stochastic dominance perfectly works. But it's not about medians, because again, median is not defined for non-numerical data. Okay, we can have A, B, B, C, C, D, D, and say that in the middle it is C. Okay, maybe this way, like in an ordered sequence, maybe this way, but not for means. So one may say, okay, Adrian, so let's stick to quantiles, not rank-based method, maybe quantile regression or test of medians. But again, comparing means is not comparing medians. They are similar only for the Gaussian distribution. So they, by definition, they overlap, they are the same. Not for other distributions. You may have equal means and different medians, and vice versa, and the opposite. So when we change the planned statistical test to something non-parametric that will change the null hypothesis, what do? We answer a question that was never asked. We answer a different question. And now the question is the new question compatible, consistent with our original problem, our original research question or not? So this is all up to us as a researcher. Luckily, there are methods that are distribution-free, semi-parametric, or actually, hard to qualify them. Let me tell. Bootstrap testing and permutation testing, in general called together resampling, right? So for bootstrap or for permutation, for example, permutation Welch t-test, both tests keep the original null hypothesis about means because we can actually compare means, but we get rid of the normal hypothesis. But so we speak about the parameter of the normal distribution, but we do it in a distribution-free manner. So I would say, okay, this is distribution-free. I don't say non-parametric. I say distribution-free. And so permutation ANOVA, permutation t-test, permutation Welch t-test. Because once I was called by a statistician who told me, you know, Adrian, for permutation testing, when we randomly shuffle observations between two groups, when we permute them, we eliminate any source of difference. So you cannot have different variances. This doesn't click with the assumption of being exchangeable between the groups. So you cannot use Welch, you cannot test means if the variances are different. But actually, because what may happen? It may happen that the means will be similar, but because of the different variances, you will get false findings. So the type level error will be elevated. But about 20 years ago, a statistician surname Jensen, sorry, I don't recall his name, Jensen, wrote a series of papers about the adaptation of the Welch T statistic to the permutation. And we have in R, we have a version we have implementation of it in the package MKinfer. And you will find the implementation of the bootstrap t-test and permutation Welch t-test. So actually, by embedding the Welch into the permutation framework, you keep the type level error on the desired level, and this works pretty well. So we don't need to worry anymore about this very case. So we have the permutation testing, the bootstrap testing. And again, I need to warn because bootstrap is usually used to calculate confidence interval. When we want to perform testing, we will need to appropriately shift means in both groups to work under the null hypothesis. This is well described over the internet. If you, for example, on the Stack Overflow forum, if you Google for bootstrap, how to bootstrap tests and shift means, just those keywords, you should be able to pretty easily find an explanation how to do this to obtain correct p-value under bootstrapping the difference. And we have one more technique. It is called G estimation. G, I mean
Yeah.
GEE, generalized estimating equations. This does exactly what the generalized linear model would, only it doesn't make any distributional assumptions. And we provide the anticipated conditional distribution like binomial gamma or Gaussian, but only to set the mean variance relationship, because it uses the information. It is based on the first two moments and needs the information about the mean relationship. And that's all. And since the special case of the GLM is the linear regression, so using the G estimation, we get rid of the normality of the residuals to make proper inference. So this method will keep the original null hypothesis if we use this method, this model to test hypothesis, but also at the cost of less efficient inference, right? Because here the inference is asymptotic at infinite degrees of freedom. And this is sometimes, less acceptable. And this also uses robust sandwich standard errors. But at the same time, we don't need to worry about divergence, from the theoretical normality of the residuals, again, as long as it is justified. So if you are convinced that it should be okay only just by bad luck, your sampling caused some skewness in the residuals on some fat tails, not much extreme, and you accept this, then fine. Then you will be guaranteed that the inference should be fine. And the last one method, this is more advanced. Professor Frank Harrell, again, he wrote a series of papers, articles, blog posts about applying the ordinal logistic regression to numerical data. Actually, it turns out that Mann-Whitney or Wilcoxon test and Kruskal-Wallis are exactly 1-to-1 ordinal logistic regression with a single categorical predictor. But one may ask, Adrian, but how does this differ from the rank-based methods? That's not all. So you apply this to numerical data, and the ordinal logistic regression will estimate a set of intercepts per each observation. And it can handle the implementation that Professor Harrell made in the RMS. This is abbreviation from Regression Modeling Strategies, RMS.
Yeah.
And his implementation can handle many thousands of numerical values, and he uses the intercepts to estimate, to recreate empirical distribution function of the outcome, of the difference, for example. So having the CDF, you can estimate from this the mean or quantize or median. So he provides, and through Bootstrap, you can also obtain the standard errors. So, and he, on one of his pages, he provides an example how he made distribution-free inference about means, but using the ordinal logistic regression in a non-standard way. So statisticians have tools to deal with difficult distributions if they really want to do means, compare means. But if people are just randomly, jump from means to medians, which is not the same in general, if they switched from t-test to one Mann-Whitney test, believing it compares medians or it compares means, this may end up with a big confusion. And I know at least one case, one confirmed, when a person we always should draw the data, visualize the data, look at the data visually, picture them. And he didn't. And he didn't look at the descriptive statistics. He just looked at the result of the Mann-Whitney test, and it was very low P, and he published. It was a very low impact factor, so it wasn't much important paper. But after years, when he read my post on LinkedIn, he messaged me and said, Adrian, I just realized that I had very similar medians, I had very similar means, but the data were just different matching distributions. The man Whitney rejected, but I reported that there is a big difference between the central tendency. And now I feel like a fool. And you know what? I cannot retract the paper because it wasn't even a journal, it was some resource. It is now inactive. I cannot contact them, but this paper goes over the internet and people may get confused by myself. So this is, what may happen. We can catch this. If you look at the data, you will see that there is inconsistency between your medians, your means, your distributions, and what the rank-based test reported. But you need to be careful. So definitely, it's not that no problems. There may be even more problems if we blindly apply non-parametric methods and we don't think what they change, how they change the hypothesis, and this way, the original questions.
Great. Adrian, I think we have come to the final question now. So what advice would you give to, biostatistician aspirants, or let's say just statisticians starting out? What topics they must, read or master? Of course, all of the things that we covered in the podcast could be the list itself, but if you just want to list it out quickly.
Yeah, if it's for newcomers, they need to start somewhere. It is impossible to embrace everything.
Correct.
And I could say this way. First, definitely descriptive statistics and methods of visualizing data. But I don't just mean learning about mean, medians, and so on. I mean study them. What I mean by study? Do some simulation and look how they behave. How a median can be changed just by single observation near the median, in the middle. So if you have 0, 0, 0, and then 100, 100, 100, one more 0 makes 0, one more 100 makes median 100. Many people don't. This is trivial fact, but many people don't expect that a median can change by several orders of magnitude just by single observation in the middle, by the frequency, not outliers, but frequency. Definitely descriptive statistics, visualization, histograms, how we may be fooled by the histograms just by changing the width of the bars, right? So of the ranges. Because for just different number of bars, we may either miss some important pattern, or we may exaggerate, completely unimportant noise. So this is important for histograms. How box plots may fool us. So we may have completely different distributions and ideally exactly the same box plots. There are examples in the internet when it is discussed a topic, a kind of plot called rain cloud, like raining, right? Rain cloud plot. And this plot, by the occasion of introducing this plot, the author shows how different distribution can result in exactly the same box plot, which may be very misleading us. So descriptive statistics and techniques of basic visualization of the data, because this will be the most important thing. And knowing lots of advanced technique, I never start without the very basic things. So they are not to be taken lightly at all. Another thing, I would say principle of inference. I know this will be painful, but again, confidence interval, estimate, what is estimator, what is estimation, estimator, what is the techniques of estimation, maximum likelihood estimation, OLS, or ordinary least square, what is the confidence interval, p-value, and so on, what is the point estimate, interval estimate, so the confidence interval, what is the standard error. These are building blocks. Without them, we won't go any further, unfortunately. Knowing the basics of the principles of the inference, I would say basic statistical tests. There are lots of them. A colleague of mine on LinkedIn, Vincent Isos, he collected over 900 statistical tests. I contributed to this list, but he did majority of the job. So about 900 of the tests. We don't need most of them.
Yeah.
But again, there is some closed in common set that we find in the literature, and it would be good to again go through them and learn, because this Mann-Whitney-Wilcoxon, chi-square test, t-test, test for two proportions, those things are basic, seem basic, so they seem easy, but they have many issues under the hood, quirks, I don't know how to say it better, issues that we need to understand to use them properly. When we know this, definitely basic modeling, but not just limited. Many people would say, Adrian, linear regression. No, GLM, generalized linear model, because it will be much more knowledge to embrace, to learn, but this covers so many topics. I mean, not the topics, but so many variants: linear regression, logistic regression, Poisson regression, negative binomial regression, gamma regression. And knowing this will automatically prepare us to work with very different kinds of data. Not only that, knowing the GLM, mastering the GLM, will enable so-called model-based testing hypothesis. Because every test, most of tests, not that every, most of tests we know, somehow uses some kind of model. On my GitHub, I proved, for example, that the chi-square test for 2 by 2 contingency table is equal to the logistic regression with a single binary predictor, to the RAO test, actually RAO score test of it. And I'm proving now more and more complex cases, including the Mann-Whitney and the ordinal logistic and so on. But also the ANOVA for comparing means. Actually, this is big procedure, and this is my next proposal is to learning first ANOVA as a procedure of comparing means, and then learning how the ANOVA uses a model, because it is not a test and it is not a model. It is procedure that uses a model and uses a test, one F test for each factor that we analyze or their interaction, and it uses a model. Why did I say this? Because the idea of the ANOVA can be much generalized, because people see ANOVA like comparing means. And I just said, I previously said that the generalized linear model is about estimating conditional means, just link transformed. So we can ask what actually ANOVA does. Next to comparing means, it turns out that it assesses something that is called mean effects. So how a multi-level categorical factor, how those levels of the factor together affect the response. So we don't focus on individual levels, groups, but how together this factor, for example, sex, not males, females, but sex together affects the response. This is what ANOVA does, actually. So we say in one way ANOVA is sex affects the response. In two way ANOVA we may have sex, we may have treatment, education, we may have race, and so on.
Yeah.
Right?
No, I said any treatment like education, yeah.
Yeah. And then it turns out that when we, especially psychologists and sociologists, they use the term mean effect. So the mean effects, ANOVA, assess mean effects. And we can do the same for logistic regression, Poisson regression. Sounds complicated, but practically it says, okay, does gender or does race or does treatment affect the counts of something? Does it changes the fraction of successes? Does it change some log normally distributed value and so on? So what we do with ANOVA can be generalized to many different models. The same can be in survival analysis. Does treatment affect the survival probability, for example? So this opens a big, but this will be complex topic, not for beginners, I'm afraid, but they can make a note to return to this topic. So while they learn about linear regression, about the classic ANOVA, please, if you listen us now, don't forget that ANOVA is a special, the textbooks ANOVA that you will learn is just special case of much general method, and you can put any single model to this, and this way you can, for example, bypass some assumptions. For example, ANOVA requires you to have equal variances in each group. What will you do if they don't? You can, instead of using the OLS estimation, you can use GLS, generalized least square, and you can address it. You don't need to switch to non-parametric methods. So by mastering this concept, this quite complex idea in the first place, as it may seem, you will be able to act more, I would say, you will be able to manage easier. You don't need to switch to non-parametric method, which may be more complicated to report, to understand, to embrace, which will potentially change your null hypothesis. And the last thing, besides modeling and the model-based testing, would be causal inference. I undermined it for a long time because I never had to work with this before. A few years ago, because this work was done for me by physicians. They decided which variables should be used in a model, what should be used, what not should be used. Only recently, I started realizing how important is to understand the role of the variables in a model, of finding relationships or trying to imagine relationships between the models. And when I discovered DAGs, directed acyclic graph.
Yeah.
Yes. So the DAG was this big aha moment to me. And from this moment, when I also learned about confounding variables, collider variables, mediator variables, moderator variables, all this completely changed my way of understanding experiments, because now I more consciously read the models and I can ask the scientist that comes to me and asks, listen, is this variable, what role does it play in the model? How can you explain this? And sometimes they are surprised. I used stepwise regression and it just returned to me this set of variables. Oh my, stepwise regression. Stepwise regression is an automated method. It just reads the data and just report what it found in the data with many possible spurious relationships. But what is your understanding of this? And when the researcher starts thinking about, he says, Adrian, it is a nonsense, a complete and utter nonsense. I need to reconsider it myself. So, although the causal inference is still something that is not commonly taught, and this is a big topic itself, I would definitely recommend to start, even if this sounds difficult, even if this is complex, because all the topics I said will be complex in the beginning. But you will start completely differently looking at the relationships, at the models, and so on. So this would be my advice.
Yeah, I think it is. Yeah, I think it's befitting that you summarized and ended with causal inference. This particular podcast is titled The Casual Causal Talk, and we also run a course on causal inference. It is catered towards marketing, but we do cover all the fundamental basics of causal inference, DAGs, confounders, mediators, etc. Adrian, so much thank you for your time. I think we've come to the end of the podcast. Even for me, this was really educational. I don't know whether you know about this. I think somewhere, near Switzerland, they have this repository of, instructions on how to rebuild the world or civilization if some cataclysmic things happen. I would definitely recommend that, this particular episode, goes into that list, because this will be required for anybody to, rebuild the civilization. The amount of insights that you have, shared with us, this will definitely help anybody to rebuild the civilization going forward. Yeah.
Thanks.
So thank you very much. Yeah.
Thank you very much, for the opportunity to be here, to meet you, to be here to talk to our audience. So it was a very nice experience, and I hope we'll have the opportunity, to meet in future again, and we can, continue this exciting discussion. So thank you very much, and see you soon.
Yeah. Thanks, Adrian.
Okay.